A very good morning to Honorable Director Sir, Professor Lalit Kumar Awasthi, the Chief Guest, Professor Amit Patra, the Head of Department of Instrumentation and Control Engineering, Dr. Rajay Singla, the faculty members and deans of NIT Jalandhar, the distinguished speakers of this STC, and dear participants. On the behalf of the organizing team of MIPS 2020, I, Dr. Karan Jain, welcome you all to the first session of day 2 of this online STC entitled as Modeling and Identification of Physiological Systems. The first talk of the day is scheduled by Dr. S.K. Pahuja. The talk is entitled as Physiological Control Systems and Feto-Maternal Monitoring. So let me briefly introduce Dr. S.K. Pahuja. Dr. Pahuja is an associate professor in the Department of Instrumentation and Control Engineering, NIT Jalandhar. His research interests include feto-maternal monitoring, electrical impedance tomography, physiological control systems, mathematical modeling of physiological systems. His research interests also include signal processing and control system. For last few years, he has been working on feto-maternal monitoring and physiological parameters measurement. So, I welcome Dr. S.K. Pahuja. So, please address the audience and start the proceedings.
Thank you, Dr. Jain. Now, good morning to everybody. Now, I am going to present the mathematical modeling of, we can say, the feto-maternal monitoring, and specifically, so what we do in this case, so we will have a mathematical model for monitoring of the blood flow from the mother to fetus and fetus to mother. Since you know that in the anatomy of the heart, in a human body, it works in association with the lungs. So because lungs is the main purifier. This deoxygenated blood is converted into oxygenated blood. But in case of feto-maternal monitoring, since lungs are not fully developed, so the full blood is from the mother side. So you can say, so whatever is the blood from the mother is the temporary organ, maybe here just like a lungs, basically respiratory system. So based on that, I will be dealing with the feto-maternal monitoring, why it is important, why the mathematical modeling of this particular system is important. Now, yesterday there were 2 good lectures based on your respiratory system and the cardiovascular system. So let me start with some sort of, you can say, a questionnaire for you, so you can see. I am giving the definition. Just listen. Definition: it is a fluid-filled network of tubes, or you can say blood vessels, through which the material moves between the environment and the cell. And what are the characteristics? It connects all parts of the system that allows individual cell or cells to thrive or take some. It's an entirely closed system, you can say. So whatever we have in this case, it is a simple closed system, and a flow is there. And what are the functions? Transport materials needed by cell for its working, just like oxygen, glucose removal, waste material, carbon dioxide, etc. like this. So this was discussed yesterday. So this system is nothing but the cardiovascular system. Karan, everybody is good?
Yes, sir, your voice is audible. You can see the screen. There is no issue. The dear audience, if you have any issue, please drop the message in the chat window. Sir, please continue.
Okay, thank you. So in cardiovascular system, this is a cardiovascular system. See, if it is not written cardiovascular system, now these are the definition, we have characteristics and the function. So what we do, what we think about this is nothing but a simple a system which is nothing but a closed loop system, and where we have some fluid and this fluid will be taken, means some nutrients will be taken by the cell for its working. So how to simulate this? This was discussed yesterday. So this is nothing but a cardiovascular system. So in a human body, it is a cardiovascular system, and in the cardiovascular system, which will be taking the help from the, what is the next? See this next. The cells of the body need energy for their chemical activities, means for improvement, for betterment, for its working, for life, you can say like this. So this energy is derived from the chemical reaction, which can only take place in presence of oxygen. What do you mean by oxygen? When we inhale, it is oxygen. So the main waste product of this reaction is the carbon dioxide. This was discussed yesterday by some professor. So what the name of this particular system? The system is a respiratory system. You can say it is a respiratory system. So cardiovascular system and the respiratory system will work together. Will work together to give you one output. The output is the good blood, you can say. So we have one waste material and one another, the bridge between this. So deoxygenated blood will be converted into oxygenated blood with the help of this particular respiratory system. See this respiratory system. So we have a, see how it is formed, means we have a one system, second system, third system, fourth system. So it is nothing but a simple unit, you can say. It's a unit. One unit is sufficient for its functioning. And second unit, we have a second unit. So both will be there to perform one task, you can say. So what is physiology? So physiology means physics and biology, means physiology, the branch of science dealing with the basic function of living organism. So first of all, we should define the basic structure of the human body. So how it is formed. So basic structure means, see this we have cellular level, atom level. So it is cellular level is the main cell, which we cannot see through our naked eyes. But cell consists of a membrane. We have an inside fluid, we have outside fluid. It's a sort of RNA, you can say like. So membrane is just like it's a semi-permeable membrane. So it will allow only whatever is required. So you can say cell is the minimum, the smallest particle, and then we have a tissue, then we have organs, and then we have a system. So first cell and different tissues will be forming a different organs. So organ, you can say, is one system, second system, third system, fourth system, first unit, second unit, third unit. So organ, different organ, just like a respiratory system, cardiovascular system. So there are different organ system in a human body. So it's a complete structure. So we can say this from the cellular level to tissue level, different tissues will form the different organ system. So just like in a heart, we have a various muscles, various tissues to perform the task. So you can say this particular cell, tissue, then it's an organ. This organ and a complete human body. So this will work. This human body will work when all units are working together. All units are independent. They are independent, but they, for a particular task, they will unite. So what is the difference between the physiological and the engineering system? You can say the engineering control system is designed to accomplish one task. And physiological systems are versatile and capable of serving different functions. In an engineering control system, it is a well-known problem, you can say. But in a physiological control system, it is considered a component, or you can say stochastic system. But will work for a betterment, you can say. The last time a word was used, homeostasis. But in a control system, we say it's a set point, but in a human body, it's a homeostasis. Homeostasis means it should be within the range, just like it's the temperature of the human body. The temperature of the human body between 37 degrees centigrade to 41 degrees. It's a range of that particular. If it is less than that, it is dangerous. More than 41, it is very dangerous. So it should be within the range. If it is within the range, it's a homeostasis. Means your system is good. Just like a blood pressure, we have a 120/80. It is within the range. So all parameters should be maintained, and this is nothing but it is known as homeostasis, or nothing but in a set point. So physiological systems are adaptive, and physiological systems have an extensive degree of cross coupling, you can say. Many things can be maintained through hormones, some other functions like this. So it is a well coupled network. So the anatomy is the study of the structure, just like what is the anatomy of the heart, and the anatomy of the physiological system, anatomy of the respiratory system, and then physiology is the study of how this body will work to perform one function. And you know the respiratory system, you know the cardiovascular system, a one unit and its anatomy and its functioning. And the respiratory system, its anatomy and its functioning. So the relationship between the anatomy and physiology will give you the correct output of that particular system. So now, see, the last time I discussed the cell, the smallest living organism thing. Then from cell to tissue, then organs. First is the cell, then it is a tissue, means tissue will have a different characteristics, and these tissues will be helping in performing or making the organs, you can say. Different organs will have a different tissue layer, you can say. All the body organs are grouped together to form a one system. So they are independent, but they will work for a betterment of the human body. An organ system is group of organs that operate or cooperate to accomplish a common task. Or you can say common task is just as a homeostasis, which should be within the set point, homeostasis. See the major organs in the human body. What are the major organs? Just like a skin, the first one is skin, respiratory system, then we have nervous system, the skin, the outer layer, the skin, then we have a nervous system responsible for all type of the controlling mechanism, you can say. On the tip of this particular finger, or on a finger, we have a different nerve endings. We have nerve endings, and these will be sensed, and the signal is approaching toward the brain through spinal cord. So there is one mechanism. The nervous system will detect that signal and will give you the output feedback signal. The cardiovascular system, you know, the cardiovascular system is responsible for producing giving the, or say nothing but a pumping system. So in the last slide, I showed you three definitions for electricity like this. So within that, we can model that particular system. So what is that in a cardiovascular system? Respiratory system. See, cardiovascular system is nothing but for blood circulation. And respiratory system, converting that or converting a raw material means deoxygenated blood into oxygenated blood with the help of the respiratory system. The skeleton system, our structure, muscular system, muscles, tissues, the endocrine system, lymphatic system, digestive system, our digestive system, then urinary system and reproduction system. So different systems, different organs, they are independent, and but at the crisis they will work together to give you one output. The output is nothing but a good homeostasis, the human well-being, you can select. See, I'm giving this example of your nervous system, the central nervous system. See the central nervous system, spinal cord, the brain. The signal from this is nothing but the peripheral, the afferent neuron. Afferent neuron means the signal, outside signal. The signal sensed by the sensor. Just like I told, this finger has 1800 sensors or nerve endings. Let's just say temperature sensor like this. Eyesight, eyes, nothing but a one sensor. So you can say the afferent signal will be sensed, or afferent signal or afferent neuron will give you the signal, and the signal will pass or will have a one you can say flow. This flow means we have a sight, hearing, smell, taste, touch, baroreceptor, chemoreceptor, baroreceptor, or photoreceptor. These are the inside our heart. So these are our sensor. Whenever temperature, the temperature will be sensed and the signal will be given to the brain through spinal cord brain. So there will be some output. This will process the signal, will give you one output, that is known as motor neuron. Motor neuron means different signal. So for feedback signal, we must have one input signal. That is input means afferent signal. And mostly the 99% the brain rejects that signal. It's an arbitrary signal, and they will try to maintain its posture like this. So let's see, whenever we are sitting like this, so we are pressure, it is a pressure. So the signal again being sensed by your brain, but it will ignore, because we can change our posture as and when it is required. See this in a nervous system. What are the parts? central nervous system. The peripheral nervous system is your sensor, brain, spinal cord, consisting of all types of nerves outside the brain and the spinal cord. So it's there for the input signal. So it will be sensed and the output, and it reaches to the brain, and there will be some output, a feedback mechanism. And sensory efferent or efferent signal, sensory neurons, efferent, and we have some motor neuron feedback signal. For this efferent signal, whenever there is a efferent signal, will be approaching to the brain, there will be some efferent signal for correction. The sensory nerves, the input signal to the brain or spinal cord, and we will have output. See this, the brain will process the signal and will give you the output. So it's the structure of, you can say, neural network. It's a neural network. So whenever, so it's a, how it works. What is the working of this particular flow of information? The flow of information from the outside input signal to the output signal. The input signal means nothing but the efferent signal, and we have a output is the efferent signal. So it is the concept of neural network also. See, the basic aim of flow of information is through. So axon means we have an input signal, will be sensed by the system, and because of that, this will generate one electrical signal. That is what is axon. Unit is axon, and this electrical signal will pass through the different layers like this, the different branches. They are known as dendrites. This is dendrites. So one neuron will have this cell body. Your cell body, the information will be gathered by this branches, this is known as dendrites, and the information will be gathered and will be given to this particular nucleus, and this nucleus will give you one output. That is, the output is nothing but an axon. Axon is nothing but electrical signal. So electrical impulse will be generated, and this electrical impulse will be passed through this particular synaptic knobs. These are the synaptic knobs. So in synaptic knobs, you can see this, the structure of the synaptic knobs. The synaptic knobs means we have a different material inside this synaptic knobs. And what is that synaptic knobs? We have a neurotransmitter. Neurotransmitter is responsible for production of the signal. So electrical signal, this is this electrical signal, one form of coded form signal is passed through or will be transferred to the synaptic knobs, and this synaptic knob, there will be a release of neurotransmitter, chemical energy. So means electrical energy will be converted into the chemical energy through this synaptic cleft. There is a gap between this dendrite and this particular branch. There is a gap, and there is this gap and this transfer of signal. The transfer of signal through the next dendrite means the next neuron, you can say, like next post neuron. So we have an input signal. See this particular snaps. So we have a nerve impulse, the electrical signal, action potential. We have electrical potential. Then we have this electrical signal is converted into chemical energy. We have through this dendrite. See this. This is nothing but electrical signal, nerve impulse. This one is nerve impulse. The signal will be given to the synaptic knobs, having the neurotransmitter, chemical energy, more action potential, more chemical energy. More action potential, more chemical energy. And this chemical energy, release of neurotransmitter, means the action potential or electrical signal generated by your axon or will be converted to the chemical energy, the chemical signal, the release of neurotransmitter. And 4 and 5, you can see the 4 and 5, this 4 and 5, the neurotransmitter receptors, there are some receptors toward the postsynaptic neurons, neurotransmitter. This will help to catch this particular neurotransmitter and will pass to the next level of the neuron, you can say like this. They cannot pass through like this. There are some helper. They are helper. They will help to pass this neurotransmitter, chemical energy. So other cell body means this particular, after this will pass through the postsynaptic postsynaptic this particular structure. Cell body, the cell body, and this electrical signal. And this electrical signal means when this electrical signal is given to the synaptic knobs, a neurotransmitter will be released, and this neurotransmitter will give you the some action potential or this chemical energy, and this chemical energy being gathered by your dendrites, and this dendrites will convert into a, and this dendrites will give the signal to the nucleus, and there will be some another axon. That is why it is chemical energy to electrical signal, and nerve impulse for the other activity. Like this is the function of this particular neurotransmitter. So everywhere we have like this. So this action potential is for every action we need some action potential. We need some feedback signal from the brain, and it should be within the range. It should be maintained. If this is disturbed, your system will be disturbed. Just like if the signal is in the range of 20 to 30 microvolt, if it is 100 microvolt, there will be a disturbance in the brain. It may be a case of epilepsy, like this. So these are the this action potential. The action potential will be generated by the cell, you can say. So at rest, whenever there is no working, you can say at rest is the minimum voltage -70. There will be no action, you can say. When the action is required, this should increase and will pass to this 0 point and will go to some positive point. So it's a rise voltage, depolarization. When the function is achieved, when the activity is achieved, it will try to come to its original position, at resting point. So at rest, there is a -70 millivolt to -90 millivolt, depending upon the different literature, you can say. It is -70 to -90 millivolt. Next, we have the membrane begins to depolarize. Membrane means the cell membrane. The cell membrane means inside fluid, the outside fluid. Because of the difference of this concentration will give you the membrane potential. This membrane potential will try to simulate and will depolarize. Will try to maintain and will rise. And because of stimulus, the membrane voltage began to rise up to plus 30 millivolt, plus 70 millivolt, like this, depending on the action. And this is done by the number of neurons, you can say. It is not because of the one neuron. It is the number of neurons will give you this particular task. The membrane volt start to means down. There will be downfall when this plus 30 millivolt has achieved and has performed its function. See, repolarization. This is a repolarization, and the membrane voltage return to its resting point, and then will stop. It's a complete cycle. Resting point will rise, then fall, and then the resting point. So depolarization and repolarization. So it should be, I told that it should be within the range. It should not be disturbed. So organ system do not work in isolation. Instead, they work together to maintain its life, to maintain its boundaries, you can say, to maintain the homeostasis. The boundary means maintaining boundaries. So every living organism, any system, must be able to maintain its boundaries. Boundary means we have a cell membrane. Inside fluid, a minimum cell, the smallest particle, smallest living organism, a cell, a membrane, inside fluid and outside fluid. And inside fluid, outside fluid, if it is maintained, you will have a resting potential, resting action potential. If it is disturbed, then there will be some action potential. So what are the, see, you can think about a cell is immersed in some fluid. A fluid is nothing but you can say it is a soup. Whatever is required from the soup will be taken by the cell membrane, and what is not required will not be taken care by this particular cell membrane. And what has to be removed, it will be removed by the cell membrane. So if it is maintained, then your boundaries are maintained, your structure is maintained. So see this, the cell membrane, how do you maintain cell membrane? Inside fluid, we have some concentration of ions. Outside fluid, we have some concentration of ions. If it is same or at equilibrium, then there will be no disturbance. Whenever movement of sodium outside is more, when it is going towards the inside, there will be a disturbance. There will be a disturbance because by the disturbance, sodium will generate the positive potential. So means the negative potential will be converted into positive potential. If it is within the range, it is okay. If it is more and more sodium is going inside, it will disturb the action potential. That way the boundaries are maintained. Every living organism must be able to maintain its boundaries so that inside remain constant. So every cell to be in a body surrounded by the cell membrane. So there are different examples. So homeostasis describes the body ability to maintain the relatively stable internal or conditions, even though the outside world is continuously changing. So inside should be maintained. Inside means 37 to 41 degrees centigrade. So this should be maintained as a homeostasis. So how it is being maintained homeostasis? If it is within the range, it is again in a control system, it is a positive feedback system and a negative feedback system. So your homeostasis is maintained with the help of the positive feedback system and the negative feedback system. Mostly in the body, we have a negative feedback system. So what are the examples of positive feedback system? In a positive feedback system, it is a stoppage of blood, you can say, blood coagulation, you can say. Whenever there is a cut, it's a positive feedback system. The birth of a baby is a positive feedback system. So I will explain the fetal metal monitoring, the blood flow, and so it is maintained. To maintain the homeostasis, we need, there are methods are positive feedback system and negative feedback system. Again, same as the control system technology. See, now come to our main point, this particular. This is the fetus. It is clear to that fetus. Now, any doubt up to this point? Karan? Any doubt? Any question?
Hear you.
Okay, any question till homeostasis?
No, sir, no question at the moment. Audience, if you have any question, please drop in the chat window, or you can directly ask Professor Pahuja.
Yeah.
We can have Q&A session just towards the end of the talk.
Okay. So next, okay, thank you.
We can have
next. This is the particular the fetus. You can say like this. So our aim is to. I will explain this particular structure. So here in this case, we know the reproductive system, and you can see a particular fetus inside the mother uterus, and in uterus we have even balloon is there. So you can say this particular is a rock. You can say like this rock, this part, and we have an inside fluid. So this fetus means inside the mother will give you some physiological parameters which we should monitor throughout the pregnancy. And you can see the structure, the growth of the fetus, start and the end point, you can say like this. So this, from the start to end, we have to monitor, and we have to convert into a model, because we have to monitor the feto-maternal monitoring with the help of some instrument. And what are the instruments? One is the ultrasound and second is the cardiotocogram. These are the two standard available instruments for measuring instruments for monitoring the fetus growth. Fetus growth, placenta size, fluid. This is nothing but this placenta, umbilical cord. And why we are interested in this particular topic, I will explain. See this. This I will explain later on. See this particular structure. See in this particular heart anatomy, you can see the blue portion and you can see the red portion. This is the working of the human body, heart. After birth, this is the working of the heart. And this working means we have this bad blood. The deoxygenated blood is converted into oxygenated blood. Deoxygenated blood into deoxygenated this blood. See this particular, the blood from this side to this side. Superior vena cava and inferior vena cava. We can say it is blood vessels or some pipes. After consumption by the cell or any tissues, the deoxygenated blood will be entering into this particular structure, this particular chamber. This you can say it is chamber, nothing but we can say it is a capacitor, storage element. And through this wall, to see through this wall, the blood is going toward this particular chamber. Again a capacitor or storage element. Through this particular structure, through this wall, the blood is going toward the right side and the left side of the respiratory system. Left side, the lung, and the right side of the lung. And after purification, the blood is entering into the left side, right side is entering into chamber number 3. Chamber number 3. And then from this particular wall, chamber number 4. And chamber number 4 will give you the blood through this particular aorta to the complete body structure. This is the anatomy of the or working of the heart, a human body heart. So it can work, it can pump. You can see the pumping. There are two pumps, first pump, second pump. The first pump and second pump. You can see this. And these are the particular muscles for generating the ECG waveform. Since we are not interested now, this ECG waveform. So what are the methods for monitoring fetal maternal monitoring? It's ultrasound and the cardiotocogram. You can see the ultrasound will give you some parameter, imaging parameter, fetal positioning, percent of growth like this and not any anomaly. And even through this particular, we can have some physiological parameter. And the cardiotocogram will give you the fetal movement and the uterine contraction, fetal heart rate. See, you can see this particular patient or this particular patient. Both are admitted in the hospital. So we can have, so whenever we need or we have to monitor the various physiological parameters, the patient should be admitted to the hospitals. So we have to monitor, see, our aim is to monitor the continuously the fetal maternal monitoring from the start to the end point. So we have another alternative. See this particular, so if you are measuring heart rate, movement, contractions, blood flow, even amniotic fluid, so it's based on 2-point system. We have a 2 points and maximum maybe a 10 point. On 10-point scale, if it is a maximum, so we can say it is a good point. So if we say, let's say 2 point like this, maximum 10 point. If the amniotic fluid is very less, okay, we can remove 2.8. 8 is good point, but you will say it is 8 is good, but 8 is a bad point if magnetic fluid is very, very low. Because if there is no magnetic fluid, it is a dangerous to the human body or fetus, rather you can say. Fetus may die. So that is why we cannot think about or we cannot say that the 10-point system is a good. So we need to monitor the feto-maternal or fetus throughout the pregnancy, throughout the 9 months, you can say like this. If we say it is, if we are removing 2 point for this fetal, this amniotic fluid, so if you say it is 80%, it is good, but it is a very, very bad. If amniotic fluid is low, it is also bad. If amniotic fluid is more, it is also very bad. If amniotic fluid is low, so immediately delivery is needed, or some action, another action is required. We cannot say. So our aim is to monitor the feto-maternal monitoring throughout the pregnancy. You can say it is required. It is essential. So we can say the now come to this particular. I will explain this cardiovascular system. It is known to us. The working of this particular heart, anatomy of the heart, the superior vena entering into the right arteria, right ventricle, left arteria, left ventricle. You can say from left side to the right side, left side to right side. See this particular lungs, right side, left side, left side is going, and then the blood is going this. So it's the working of the actual human body. You can say after birth. But what is before birth? What is before birth? What is after birth? You can say the lungs are fully developed, so it can work. It can give you the oxygen. Your deoxygenated blood can be converted into oxygenated blood through some alveoli. You can say like this. The structure. See this particular structure. It's a after birth. Here you have a cardiovascular system, right side and the left side. Again, it's going towards the respiratory system, and the blood again coming to the left side and consumption. It's a human body structure after birth. It is after birth. Your lungs are fully developed, which can convert your deoxygenated blood into oxygenated blood. So when we inhale, we inhale And then this inhalation means we are converting a oxygen into, means oxygen will be given to the blood and the carbon dioxide will be taken by the blood. So it's a respiratory system is responsible for purification, we can say. The closed system and respiratory system for a human body. It's a complete for human body. The circulatory system, human body. See this particular, a newborn heart after birth. See this particular heart after birth and heart before birth. So means it is open. It is open now. Fetal heart before birth. Because the blood, whatever the blood from the mother side will pass through the all chambers, chamber number 1, 2, 3, 4, and like this, it will give the blood. Only the mother blood is responsible for working of the fetus, you can say like this. Because of your lungs, the lungs in the fetus are not fully developed. So after birth it should close. See this, it is closed. Permanently it is closed. If it is not closed, then it is known as the heart hole. The heart hole, when there is a gap. So in a mathematical modeling, we can take some assumptions also whenever there is a heart. Because see the purification of bad blood and the good blood. So good blood will be less available in chamber number 4 because of mixing of and the quality will be bad. So in a mathematical modeling we can take this constraint also. See this particular, so placenta. So see this placenta is responsible for giving you the blood to the fetus. That means placenta is just like a respiratory system for a fetus. Placenta provides oxygen. Even placenta takes deoxygenated blood also. So that way feto-maternal monitoring is very, very essential, you can say like this. Because in the feto-maternal monitoring, the blood is from the mother side, is entering to the fetus side. And it is monitored with the help of ultrasound machine. So in high-risk pregnancy, if we start monitoring the blood flow system, you can say like this, the feto-maternal blood flow. So it may take more time. It may be dangerous to the fetus also. So we should have some adaptive, or we should have some predictive models just to predict the flow of blood from the mother to fetus and fetus to mother. See the blood from the mother side is entering to the blood to the fetus, a good blood, and this good blood will be consumed by the all or working of the fetus. Then the good blood will be converted into deoxygenated blood, the blue one, and this blue will go to the mother side through some temporary organ, and this temporary organ nothing but is known as placenta, and this placenta is responsible for supplying of blood and removing of the waste material. You can say the placenta is a temporary respiratory system. Placenta is a temporary organ developed when the fetus start growing. Whenever there is a fetus, you can say like this. So why we are interested in this particular study? So you can say the working of this particular respiratory system. It is a homogeneous system. It is a homogeneous system, but the fetus is a heterogeneous system. What is heterogeneous system? Because we have a body within another closed path, you can say. A fetus or a baby is in a balloon, and then this balloon in a uterus. So body, heterogeneous closed system within another closed system, you can say. It is very difficult to monitor. That is why we are going for some mathematical modeling, some predictive models just to predict the blood flow between mother and fetus and mother life. So the possibility of substantial signal loss not being tried for fetal monitoring. That is why we should go for some mathematical monitoring also. So as per the literature, very good literature or survey, some journals say that we should go for some mathematical predictive models rather, you can say, fetal-maternal monitoring, the blood flow. This particular structure. See, this is placenta, a temporary organ towards the mother side, and you can say this particular red or pink is nothing but a one blood vessel having good blood, and this blood is consumed by this particular fetus, and then there are two blood vessels in a blue color for removing of the waste material. So this particular rope, or this is known as umbilical cord, and this umbilical cord is the lifeline of the fetus. You can say like this. The umbilical cord placenta is nothing but a respiratory system for a fetus. So umbilical vein. So umbilical vein is carrying the good blood and the waste material removed by the arteries, you can say. But in a human body, vein is nothing but for carrying the bad blood. But the arteries are for carrying the good blood. But it is reverse in this case. You can think about this also. Why vein is for good blood and artery for the bad blood. See this particular mother side, fetus side. It is a fetus growing inside a closed path. It is a particular placenta. It is mother side, umbilical cord, this fetus, and umbilical cord having three blood vessels also. The three blood vessels. One is for these blood vessels, one vein, two arteries, you can say. Vein is for good blood and arteries for bad blood. So what is the mathematical model? Why we are going for mathematical model? And I explain. Why we are going for mathematical model? This blood flow is being monitored with the help of ultrasound machine. If let's say in any case, in any complicated case, one artery is missing, and you know one artery is missing, since you know at the time of first check, you can say it is one artery is missing. There is only one for good blood, one is for bad blood. Double fetus, we have a two placenta and two fetus. Third one may be a two fetus, one placenta, two fetus, one placenta, and two this umbilical cord. There is one placenta. So you can predict, since you know the structure, you know the behavior of this particular system. Now you can predict you should have a predictive model, just to predict what will happen whenever there is a one blood vessel, one artery, or two blood vessels, two fetus, two placenta, two fetus, one placenta, like this. So we can predict. So the mathematical model for fetal monitoring, blood flow between the fetal circulation, taking two notes based on the Markov model. Markov model means it's a good state and the bad state. In an electronic system, we can say it's a good state, working state, and bad state is a not working state. Fan is working, fan is not working. Electronic component is working, electronic component not working. So good state, we can say whenever we have a blood, the blood from the mother side, the input, and it is how much time will be taken by this particular blood to reach the fetus side. And after consumption, and deoxygenated blood should reach to the mother side, fetus side. So oxygenated blood flow from mother to fetus, and deoxygenated blood from fetus to mother. The model interprets how the oxygenated blood flow from placenta, one node, to the second node, umbilicus. Umbilicus means this navel point, the navel point where we have the entry point of the fetus, the umbilical cord, and deoxygenated blood to the placenta from fetus. The same model we can simulate for the different conductivities. Conductivity means the path of this particular umbilical cord, because umbilical cord is nothing but some temporary structure. and the conductivity may increase and the conductivity may decrease also. To see the effect of uterine contraction, because uterine contraction is nothing but an indication of the blood flow. If uterine contractions are more, it will compress the blood vessels, you can say, umbilical cord. It will reduce the supply of the blood to the fetus. And if it happens, the ECG or the heart rate or heartbeat will be more. There may be a less blood to the fetus and there may be hypoxia. Fetus may die. And it may affect the brain of the fetus, uterine contraction. That is, it is very important parameter to monitor. If you say uterine contractions, and uterine contractions are responsible for fetal birth. Now, I have developed this particular model in a lab environment. You can simulate in any other environment also. So, now this particular structure now, fetal movement. See, this side is, you can say, placenta side. Mother side, placenta side, the nerves endings, nerves, there are nerves, blood vessels, complex system, temporary organs, and then the blood through this particular umbilical cord. This here you can see this one point. This second node, umbilicus. This umbilicus second point. I will show you later. This for second node. So we have first node here and second node. Placenta. See, this is for good blood. This is for good blood. There is one vein, and for bad blood there are two arteries. So node 1, a good blood means state 1, state 2. State 1, blood from the mother side will reach to this side, and after consumption, the fetus side, the blood should reach to the mother side placenta. Node 1, node 2, mother side and the fetus side. See, just see the anatomy of the fetal development. Just see the fetal development, how it is, how to, since you can make a good model if you know the anatomy of that particular system. That is very, very important. Then you can develop a good model. So baby develop in a uterus with a life support system for the fetus, and is composed of umbilical cord. You can say it is umbilical cord, a rope, a placenta toward the mother side, and amniotic fluid. It's very important. You can see the umbilical cord connecting the, you can say it's a blood vessel, or it's a cord from the mother to fetus. And the placenta is temporary organ for supplying good blood and for receiving the bad blood and amniotic fluid. The placenta is a pancake shape. So you can say it is a big shape and is attached to the uterus of and is connected to the fetus with the help of an umbilical cord. The anatomy. The umbilical cord is the lifeline between the fetus and the uterus. That is, mathematical modeling is required. Predictive model is required. See, if you are monitoring this cord, and this cord has ruptured, your placenta has removed from the uterus, what will happen? There will be no blood supply, and we are going to monitor like this, so we can predict model. After it is formed, it starts functioning throughout the pregnancy to protect the blood vessel, travel between the fetus and the placenta. The responsibility of the placenta is to act as a point of trade between the circulatory system of mother and the fetus. That is very, very important. There are several small blood vessels that carrying the blood from the fetus and like this I already explained. The waste product of the fetus and are transferred to the mother blood on diffusion principle. Means it's a passive system. It's not active, it's a passive system. From low concentration to high to low concentration. Diffusion means there is a high concentration towards the fetus side and the low concentration towards the mother side. And in the case of the good blood, high concentration toward the mother side, low concentration toward the fetus side. That's it. Just diffusion. It's a passive mechanism. It's very important to know the permeability of the fetus, of placenta, you can say placenta permeability and the umbilical cord and the total membrane diffusion conductance of the placenta membrane. That is very, very important. That is the predictive model is required. Permeability is expressed as the quantity of the substance that will diffuse through a unit surface that placenta membrane for a given concentration. the total membrane this conductance or the permeability times the total surface area. The permeability increases with the pregnancy. It is very important to know the relationship between the concentration quantity of the supply of the blood at the mother side, and it is how quickly it reaches to the fetus side through blood vessel means vein. And similarly, deoxygenated blood through the mother to fetus to the mother side through arteries. So there are three blood vessels. Just now concentrate on the model. One is vein, second is arteries. This cord can grow up to the age of this length of 50 to 60 centimeter. The standard size of the umbilical cord is 50 to 60 centimeter. If it is less than this, it may be a dangerous. There may chance of disconnecting this particular cord from the mother side. It may remove the placenta. If it is more than 60 or more than the required length or whatever is then there may be a knot also. Or it may tie it around the neck of the fetus also. That is the standard length is there, which allows the baby to have enough space to move safely without damaging the placenta or umbilical cord, because it's a lifeline. Without that, fetus may die. There may be dangerous. There may be oxygen less, hypoxia. The placental connectivity increases with the age of pregnancy. More and more blood is required for fetal growth. So during the time of pregnancy, it can be found that umbilical cord is in the form of knot. Form a knot. Or means there is a गांठ बन जाती है जैसे knot. It will reduce the supply. But at this time, the cord wrap around the body of the baby, or around the neck, you can say, just like a funda. So we should. That is why it is a good model is required. The blood supply may decrease, so it is necessary to know the blood profile of in the fetus. Whenever there is a knot, or whenever there is a umbilical cord is formed knot or wrap around this particular fetus, so it will reduce the supply of the blood. Mathematical model is required now. Now discuss the mathematical model. A mathematical model for the circulation of the blood in the fetus circulatory system is developed by taking the two-node concept based on the Markov model, and how the good blood, oxygenated blood flows from placenta, one node, to the second node, umbilical cord, and how deoxygenated blood from fetus to placenta, like this. The model is simulated at the various connectivities. Various connectivities, the connectivity of the vein, connectivity of the arteries, like this, and the knot, and around the neck. So different connectivity will be taken care by that. And so if you know the anatomy, what is the purpose of explaining this? This is not anatomy. If you know the anatomy, you can have various constraints. You can put more constraints, and you can have a better model. So also we tried the effect of the uterine contractions also, because uterine contraction will remove may decrease the blood supply. See this particular structure now. This particular structure we have to convert into a model. See this placenta, this way, this second node, umbilicus. See this fetal umbilicus, navel. Unni point, this navel, like this. So node 1, node 2. Node 1, node 2. This placenta mother side, this is fetus side. The blood, good blood, vein, and bad blood, arteries. So it is GV, the connectivity of the vein, the connectivity of uterine, this artery 1, artery 2, like this. So this model. So we can say good state means oxygenated blood is going toward the fetus. The fetus will consume the good blood for its activities, and then the bad blood, deoxygenated blood, will move to the fetus side. So this placenta is nothing but a simple respiratory system. Proposed model, this figure is converted into this model. The figure 1 represents the complete anatomy of the fetal circulatory system. Here, node 1 represents the full of oxygen, rich of oxygen, full oxygen, mother side. Node 2 represents the fetal side umbilicus. There are two stages. Stage 1, placenta attached to the mother side, say node 1, full of oxygen, and stage 2, umbilicus, second stage. State 1, state 2. So state 1, state 2, or node 1, node 2, node 1, node 2. Now again converted to this is now. So there are some assumptions also. Whenever we are designing some simulation study model, we have to take some assumption. Here assumption is this is a straight line. So, see this oxygen, nutrients from the mother side, placenta, vein. It is simply block diagram reduction. Umbilicus, fetal heart, and the blood is assumed by the. So this fetal heart. So we can assume this as a different. We can take different nodes also. This for node 1 and node 2. So umbilical cord between the placenta and the this. See this signal flow graph, like this. So this is nothing but node 1. This we can say is the node 1. IA, IB node 1. This is node 2 only. See this node 1, node 2. Placenta attached to uterine walls, input blood from the mother side, 1 and 2. So what is Markov model? In Markov model, so we can predict the state 2 with the help of state 1 after time delta t. That is the advantage of Markov model. So Markov model is a stochastic system, and this our system is also stochastic system. It is very easy to predict. That is why we can take the help of the Markov model. Let's say P0T, the quantity of the blood at node 1, good blood. P1 is the quantity of the blood reaches at node 2, and after consumption it will go to this particular placenta side. GV, the conductivity gain of vein. U1, the conductivity of artery 1, artery 2. So after time delta t, see this P0T at t equal to 0 at mother side will reach after time delta t to the fetus side. So this is equation. This equation you can take this equation you can write from any book, from the reliability book. It is known as probability of occurrence. It is known as under reliability. It is probability. So P0T means the blood at node 1 will reach to the node 2 after time delta t. So time it is a unit term. Okay? So P0T means, see, if we convert this into a equation, we try to find out the response of this particular system, then you can see the response. This P1T, when this blood is reaching to the node 2, it will be consumed by the fetus, and then fetus will give you the deoxygenated blood and will reach to the node 1, back to the node 1. Equation 1 and 2 are very, very important. So similarly, this we can write, you can see this particular, how to solve this particular equation. This we can solve with the help of Laplace transformation also. You can use any method. After Laplace transformation, we can go for its IR filter also with the help of impulse invariant method, match transform method, and we can simulate this equation also in continuous system and in discrete system. So here P0T, the good blood, P1T is the bad blood. So these equations, you can see the total output is like this. This equation number 1, equation number 2, the final output this P0T. P0T is the good blood from good state to node 1 to node 2. And P1, the blood, deoxygenated blood from node 2 to node 1. If you simulate equation number this and this, so how much time will be taken by the blood to reach from mother to fetus, fetus to mother by this like this. It unit time you can say. See, this we have simulated in LabVIEW. See, this is the front panel, you can say. This is the front panel. Now you can give some output. You can see the conductivity from 0 to 1. this GV, the input blood, the blood like this. So you can simulate. You can simulate again like this. You can see the output conductivity. You can see this. Increase the conductivity. If this is one is not working, see this P. If this is not working, you can see this particular. It will take longer time to reach to the mother side. When I took one example, when there is one artery, second is missing, two blood vessels. This is missing. Again, this response. You can see the response. This you can simulate. You can see this. You can simulate. You can simulate. See this. So based on that, what are the results? The software design in LabVIEW at different values. You can see when P0T. So you can see the unit time in millisecond, microsecond, but our aim is to check this particular response. How quickly it reaches the node 2 side, from mother side to fetus side, through umbilical cord, through vein you can select. If conductivity, this conductivity or this time, unit time will reduce as the pregnancy is improving, or pregnancy is progressing. So this will improve. So it should take very less time to reach it to the final value, means node 2. The response of P0T shown here with 100% conductivity, We have assumed that the vein is fully functional. There is no, and this umbilical cord is functional. There is no wrapping. See, when the conductivity is reduced, the setting time increases as conductivity reduces. You can see this particular. If this conductivity reduces, means because of any reason, because of, you can say, when say the umbilical cord is very small, umbilical cord has a knot, the umbilical cord is moved around the neck, you can say, so this will take the longer time to reach the node 2 side. Similarly, when both this is P1T, means from fetus to the mother side, and this is not when both are working. Both arteries are working. You can see when one, okay. So based on that, the factor influencing the node 1. Node 1 is to the mother side. What are the factors which may influence the supply of the blood? High blood pressure to the mother. High blood pressure to the mother will reduce the blood supply to the fetus. It is dangerous. Blood supply to the fetus will be reduced. When there is a kidney disease to the mother, it will reduce the blood supply. Placenta will not be fully functional because of the diabetes. The heart or respiratory problem to the mother, infection, alcoholic drugs. The node 1, the constraint at node 1, we can take these constraints and then we can give the blood supply. So what is the effect if at node 1? Node 1 means to the mother side. The factor influencing the node 2. I explained, see this, instead of 2 arteries, now there is 2 blood vessels. Only this is if you are monitoring this system, it will take longer time. Now you can have a predictive model for this also. Once you have monitored, once you have checked, there is only 2 blood vessels, 1 vein arteries, then you can predict. Unnecessarily, we should avoid monitoring through ultrasound machine. If you know, long cord, you can see this long cord. There is a node, long cord, and because of the long cord, there will be a nuchal cord. Node aa jayega, okay? And because of the short cord means this may rupture the placenta because fetal has to move inside the fluid. It will move, rotate, float करता है इसमें. So there are chances when this umbilical cord may remove the placenta. So prediction may prevent fetal hypoxia. Fetal hypoxia means the supply of blood is very less. That is hypoxia. If it happens, this is dangerous to the fetus. Fetal death. There is no fully blood supply, blood supply to the fetus, and no oxygen is given to the fetus. Uterine contractions are more, heartbeat is more than 220, then there may be a fetal death, low birth weight, and placenta abruption. Placenta abruption means placenta will remove from the uterus. It is very dangerous. So we should have a predictive model. And if you know the anatomy, you can predict. So basic structure is like this. Okay. So first method is the ultrasound method. We can monitor, but how to monitor continuously the movement, the placenta position through electrical impedance tomography. So I will stop here. Now, any questions, any doubt, you can ask. Quickly, I can show you the slides. Because this method can help you in continuous monitoring of the fetal movement. Yes, any doubt? Any questions from my side? I will stop for the time being. If you have any question, you can ask. The fetal movement. And before ending, I can explain. See, in a feto-maternal monitoring, first of all, we need the continuous monitoring. And the feto-maternal monitoring is generally done with the help of the ultrasound machine and the cardiotocography machine. The ultrasound machine, we need some experts, and the cardiotocogram, the patient will have to admit in the hospital. It is continuous monitoring. So fetal movement, the basic parameter is required. It can be monitored with the help of the fetal movement system. The cardiotocogram can give you the fetal heart rate and uterine contractions. Ultrasound can give you the growth. So we have imaging and physiological parameters. So for imaging, we need ultrasound machine, and for physiological parameter, we need cardiotocogram. Even ultrasound, we can have a fetal movements. But for continuous monitoring, we cannot use ultrasound machine and CTG. So we need another instrument. We need another instrument to monitor the continuous monitoring. So for that, we try to develop the electrical impedance tomography system. The electrical impedance tomography system based on the impedance cardiogram. You know the human body, the different human body structure systems, different organs have different conductivities. So different conductivities can be monitored, and very good contrast image can be formed. And this image known as electrical impedance tomography, or conductivity image, you can say. And this conductivity image, I will show you this slide. So for imaging, we need energy. And this energy is passed to the object, and it will detect, output will be there, and this output will be converted to some mathematical model to some signal, some model, this image reconstruction. But in imaging techniques, ultrasound machine, CT, PT, like, so we have some energy. But in electrical impedance tomography also, we have one energy, is electrical signal. We have a signal, and this signal will be given to will be fed to the human body, and we will have a output signal. And this output signal will be brought into this image deformation through mathematical modeling. So electrical impedance tomography is another solution for monitoring the feto-maternal monitoring continuously. So this EIT has some advantages and disadvantages also. So advantages means contrast image can be monitored. It is a continuous system. It is a continuous system. Continuously we can monitor the image or the parameter of the human body for fetal maternal monitoring. The functional images can be obtained through the EIT. The good contrast contrast involves no ionizing radiations and poor, and the disadvantage is poor and position dependent spatial resolution is required. Low sensitivity to the inner organism. These are some advantages and disadvantages of EIT, and with the help of EIT we can monitor various physiological parameters of the fetal-maternal. Thank you very much. Any questions?
So thanks a lot for the wonderful presentation. So someone has asked a question that if you can give a brief idea of Markov models, what do you consider are limitations in your model?
What are the?
So they are saying that Keshav Kant is the participant. He is saying that if you can give a brief idea of Markov models and what do you consider are limitations in your model?
See, the Markov model is nothing but it's basically a predictive model, we say. So Markov model is used in the reliability theory. So reliability means, as in the case of electronics component, in electronics component, it's a good state or a bad state. If it is a good state, it is 100% working. If it is a bad state, it is a poor state. There is no probability of that occurrence. Good state means reliability is 100%. Bad state, reliability is 0%. Probability is 0%. So let's say if fan is working, 100% is working, fan is not working, bad. So so we can predict the second stage, state 1, state 2 with the help of state 1. So for detail, you can take any book from reliability engineering, Markov model by reliability engineering. It's a very good topic because in a stochastic system, since our Markov model is stochastic system and our human body is also stochastic system. So it is a fit use of this particular Markov model. And the second question is, what are drawbacks? Drawback is, one of the drawback maybe, since it is simulated study, in simulation whatever we do, we know. And in simulation we have the 100% reliable parameters. And in this case, in our model, I have assumed that this umbilical cord is a straight line. Umbilical cord is a straight line. That is one of the biggest constraints you can say. Since it is not a single, it's not a straight line. It will move. It is a curve like this. There may be a node also. There may be a curve also. So if it is so you can assume it is a curve also, that you can have a better model also. Thank you. Next.
Another question is, ultrasonics used in physiology has a tolerance limit?
Ultrasound?
Whether the ultrasonics used in physiology have tolerance limit?
See, in a ultrasound machine, I explained in this EIT also, the ultrasound machine, we cannot go for continuous monitoring. In ultrasound, we are inserting the signal, energy. And this energy, if you are monitoring continuously, this may damage the fetus, you can say like this. So yes, it is a tolerance. We cannot have the continuous monitoring with the help of ultrasound machine. So since ultrasound machine, everybody cannot use this ultrasound machine. The doctor or expert can use. So first of all, so for even EIT is nothing but one screening tool, you can say. But we cannot replace ultrasound machine. For better result, we should go for ultrasonic imaging also. So for some limitation, for few seconds, few milliseconds, for lesser time, we can monitor the image also. We can go for ultrasonic machine. Continuously, we cannot use ultrasound machine. Yes.
Okay. So in the audience, if you have any question, you can please ask. So another question has come. Can we use MRI scanning for that?
No, we cannot use MRI in pregnancy. We cannot use MRI in pregnancy.
Okay.
There are limitations. We
Okay.
cannot use this. Even we cannot use the CT scan or like this.
Okay.
There are limitations in ultrasonic this for fetal monitoring.
Okay, sir.
So if you have any doubt, you can write. I can give you my email address. If any doubt, you can ask. We can interact, and we can have a mathematical model also. This is my email address. pahujas@the rate of nitj.ac.in.
Okay, sir. The audience, you can please take a note of this. Dr. S. K. Pahuja, sir, thanks a lot for your wonderful presentation. I
Thank
hope the
you very
audience
much.
enjoyed it all. Sir, there,
I appreciate you
yes,
listen very carefully, and I thought you must have gained something from this particular. See, the many things we can solve with the help of mathematical model. So in the biomedical application, we should go for mathematical modeling also for our research work, and then we should go for phantom studies. Since nowadays it is very difficult to get the ethical clearance for human body or you can say animal study. So first we should go for mathematical modeling, then we should go for the phantom studies, and then you can do your research. Thank you.
yes, sir. Thank you, sir. And there is an update. There is an important update, and today's afternoon session at 2 p.m. Dr. Nikhil from Shastra University would be presenting. So the title of the talk would be Physiological Systems Modeling, Simulation and Control. Please join us back at 1:50 p.m. We shall start exactly at 2 p.m. The talk will proceed at 2 p.m. But please join us at 1:50 p.m. for a little bit of introduction. Dr.
Thank you,
Bujal,
Karan. Thank you,
thank you,
audience.
sir. Thank you, everyone.
I can leave now.
Yes, sir. Sir,
Can
have a
I
nice
leave
day.
now?
Yes, sir. Yes, sir.
Thank you. Thank you.
Have a nice time, everyone. Okay,
Thank you.
join us
Thank
back
you.
at 1:50 p.m. Okay.
Hello everyone. Is the voice audible?
Yes, sir, it's audible.
Okay.
Audible, sir.
So can you proceed or we have to wait for 4 minutes? What do you suggest?
Sir, the talk should begin at 2 p.m.
2 p.m. Okay.
Meanwhile, you can
So
introduce Dr. Nikhil.
so audience is I think joined. Participants, I think 28 participants are joined.
Yes,
Okay.
Dr. Nikhil has joined. Welcome, Dr. Nikhil.
Okay.
Thank you, Dr. Jain.
So let me start the session. So
Yes,
hello
please.
everyone. Hello everyone.
Hello, sir.
I am Dr. Om Prakash Verma, Assistant Professor in Department of IC. On behalf of MIPS 2020, I welcome you all in second session of the MIPS 2020 on the second day. As a course coordinator, it is my great pleasure to introduce Dr. Nikhil Pachauri, our second plenary speaker of this series of the second day. Dr. Nikhil Pachauri is currently associated with the School of Electrical and Electronics in Shastra University, Tamil Nadu. Dr. Pachauri research and development experience includes more than 5 to 6 years in the industry as well as academia. He received his MTech degree from NIT Jalandhar. He is alumni of our institute also. So, and he pursued his PhD. He has completed his PhD degree from Delhi University, MSIT. He works in the multidisciplinary environment involving machine learning, machine intelligence, process dynamics and control, process control, physiological control system, fractional order control machine, and applied to various real world problems. He has given several conference plenary lectures as well tutorials and invited seminar lectures in many of the reputed universities. He has published several book chapters and more than 10 essay research papers in the very reputed journals. He is member of IEEE and reviewers of many reputed peers reviewed journals. So I welcome Dr. Nikhil for accepting our invitation on your busy schedule. So please, I am handing over your our session to you, or hope all the participants will get some benefit from this session. Okay, thank you.
Okay,
Doctor,
hello. Thank you, Dr. Verma. So before going to discuss the presentation, I would like to thank Dr. Verma and Dr. Jain for giving me an opportunity to give an expert talk for this STC. So here I am going to present my presentation. So
Dr. Ravi Verma, please unmute your mic. Dr. Ravi Verma.
I'm audible?
Yes, 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 Nikhil. Yes, doctor.
Okay. So, I'm going to share the screen.
Yes, please your screen.
I'm sharing. Okay, so it is shared. Everybody is able to see.
I think Dr. Karan is visible to you?
Yes, the screen is visible, but please continue presenting. Just
Okay, I got it. Okay, it is shared. So, good afternoon. So, I'm going to present my presentation on the topic physiological control system. Sorry. Good afternoon to all of you. Today I'm going to give an expert talk on physiological modeling, simulation and control. So as Dr. Verma said, I am Dr. Nikhil Pachauri as an assistant professor from Shastra University. So this is the during the course of this presentation, I'm going to discuss this what is physiological control system is all about, and the second is homeostasis, example of physiological control system, importance of mathematical modeling, and mathematical models of drug discovery and development process. Then I am going to take two examples. One is chemotherapy model and how it is being simulated on the MATLAB and blood pressure control. And also I am going to discuss how we can design a PID, simple PID controller for chemotherapy model as well as for blood pressure control. So the first question that arises in our mind is what is physiological control systems are? So as we see if I bifurcate this whole term physiological control system, so it is a combination of three words: physiological, control plus system. But generally we used to say control system as a single word. So again it can be divided into two parts: physiological plus control system. So first, what is physiology, what is physiological, and what is control system? So physiology. Physiology is the science of life. It is a branch of biology that aims to understand the mechanisms of living things from the basis of cell function at the ionic and molecular level to the integrated behavior of whole body and the influence of the external environment. So this is basically a you will say a bookish or a prescribed definition of what is physiology. If I wanted to say what is physiology to a layman language, so in one word we can say it is nothing but a study of a human body. Whatever inside the human body is going on, we study that behavior of whole human body that comes in the physiology. So this is all about the physiology. So we can bifurcate, like either it is a science of life or a biology, branch of biology, or we can say it is nothing but a study of a human body. Next term is control system. I think everyone is very much aware about the control system, but here it is important to discuss the control system because we are going to during this presentation we are discussing about the physiological control system. So little bit about the control system I am going to take up. Within one or two slides I will complete this. So control system. Control system is nothing but a collection of interconnected components that can achieve a desired response in face of external disturbance. Same. Control system is a collection of different blocks, and each blocks or each component is responsible to achieve the desired, whatever the desired output we want. So this is all about the control system. Now, if we add these two terms, physiology and control system, we get a word physiological control system. So we can define a physiological control system as the in physiological control system we can say that human body is considered as a system, and the internal environment of the system and the internal environment of the human body refers as a human body as a control. Physiological system is in physiological system human body is considered as a system, and the internal environment of the human body refer to each and everything present within the system where cell lives and work together. Or we can say that our human body is a collection of trillions of cells. So mainly cells are responsible for the proper function of the whole human body. So we have body has to maintain certain parameter at certain point so that all the cell should function properly. And one more thing in physiological control system is that a specific and optimum condition should be maintained within the body so that our cells functions properly in order and in turn which makes let's see, like our body is having tissues, different type of tissues, organ and circulatory system. So if cells work properly, if we maintain the temperature, if we maintain the favorable condition around the cell, the cells works properly, which in turn every tissue, every body part will work properly. So it is also necessary to maintain the composition and the temperature around the cell. The temperature and the composition of the around the cell should be constant. If anything, if either temperature will increase or composition will change, so it is going to affect the functioning of the cell. So in all, we can say that physiological control system, here we talk about the how to control different parameters in the body, or how body will control itself different parameters. So this is about the physiological control system. Now, type of control system. We generally know there are two types of control system. One is open loop. So this is a block diagram representation of a open loop control system. It is also referred as a non-feedback control system. So now, what is the property of this open loop control system? As we see, output is linearly dependent on the input. So whatever change in the input, the reflex is directly we can see in the output. Or we can say that, so there is no control on the output. So how the input changes, accordingly we get the output. So here is a so this is the plant which is having, if we consider it, it's a transfer function as GS. So GS multiplied by the input will get the output. So change in the input will get the change in the output. Now the drawback of this open loop system is that first is we don't have control of the output. Whatever the input, we don't know how output will behave. But yes, if we have the bounded input, then we have the bounded output. Means if we bound our input within certain range, where we know that this plant will work well for the input range between +5 to -5. Let's suppose if this plant works well between the range of +5 to -5. Any range beyond this particular, any value beyond this range from +5 to -5, then output will be abrupt. We can't predict what will be the output. So we can say that the output is stable till the input is within the bound itself. So the main characteristic of an open loop, we can say that there is no comparison between actual and desired value. And also open loop system has no self-regulation or control action over the output. Means output cannot correct or self-regulate itself. It is entirely depending upon the whatever the input values we are giving. So we have to be careful what input values we are giving to an open loop system. That's why it is always say that before going to do any analysis, you have to, before going to a closed loop system, you first do certain analysis about the open loop system, how system will behave, on what values of the input, if there is any model uncertainties, how the model parameter is going to affect the output. So these all analysis we can done with the help of open loop system. So the drawback is that it is we don't have any we don't have any control over the output. Now the second block diagram. It shows the block diagram for the closed loop control system. If I take this part only, where plant, manipulated variable and output. So this is nothing but a open loop system. So how it is different from open loop system? It is having 3 more blocks: controller, comparator and feedback. So it is also called as, so this closed loop system is also called as feedback control system, which uses the concept of open loop system in a forward path, but has one more feedback path. So it uses the concept of open loop system in forward path, and also having one feedback path, one more feedback path, or we can say feedback loop between the reference input and the output. Between the input and the output. So how it will work? Let's suppose I have to maintain the value of the output at 5. So reference will be the 5. We know that I wanted to maintain my value of the output at 5 degrees centigrade or anything PSI in terms of pressure. If I wanted to control the pressure. So what will happen? Initially, whatever the output is generated by the plant, it is feedback by this feedback loop to the comparator, where this output is compared with the difference. It generates the error, and according to that error, controller will generate a signal that signal is also called as manipulative variable or manipulative signal, which manipulates certain parameter so that the output will reach to reference input. Or we can say that controller will work to minimizes the error between the reference and the output, so that we can easily achieve the reference, which easily achieve the output value which we have set. Right? That is called the reference input. So this is the difference between open loop and closed loop system. And also there is one more advantage of closed loop system. Because of this feedback loop, the sensitivity of this closed loop system towards parametric uncertainty. What is parametric uncertainty? See, plant. If I write my plant either in terms of some mathematical equation or in terms of transfer function. So let's suppose the gain of the plant is the gain of the plant will increase. So that gain, because of that gain, there is some change in the output. In order to reduce that effect, a feedback signal is important. So it will reduce, because of this feedback, it will reduce the effect of that increase in gain at the output. So this is another advantage of the closed loop system. So sensitivity towards parametric uncertainty is less as compared to the open loop system. Next is, now I am going to discuss a very important part, that is a very important term called homeostasis. Now, what is homeostasis? What is homeostasis? It is defined as the property in which the variables of the system are regulated for maintaining a stable and relatively constant environment in the internal environment of the system, despite the change in the external environment. We can take so in order to know what is homeostasis, I am going to take one example, that how body will regulate or self-regulate or self-control its parameter. Let's suppose you are going for a walk or for a run, or evening walk or evening run, or morning walk or morning. During running and starting, when you start running, what will happen? You will not. First you will start running slowly, and gradually you increase your pace. After some times, you will feel there is a sweating from your body. Why? Because as you run, the temperature of the body will increase. So, in order to, as we know that the body temperature should be maintained at 37 degrees centigrade. So, if the temperature will increase, body or human body will self-regulate itself by opening these sweat glands, so that more sweating will be there. As much as sweating is directly proportional to the temperature. More sweating, lower the temperature. One thing is that. Second thing, as you see, after completion of running, we used to take a deep breath. So that is also a very good way to maintain the temperature, to take the temperature to its normal value, that is 37 degrees. So this is all about the homeostasis. Means how body self-regulate or self-maintain or stabilize itself, even though there are lots of external environment disturbances is there. So this is, and second good example is pressure. So the body pressure should be maintained within the 120 to 80. So the critical value of body pressure or blood pressure is 120 mm Hg by 80 mm Hg. Means maximum is called systolic and lower is called diastolic. So systolic pressure is plus 120 mm Hg and the diastolic should be 80. So if a person is having body temperature around 37 degrees centigrade, we can say that it is a normal body temperature and pressure should be 120 by 80 degrees. So next is, this is the block diagram which shows how homeostasis and body system are connected to each other. So as per the concept of internal environment of the system, all the body system are also part of the internal environment. Means whatever is going on, it is going on inside the body. So if I say internal environment, it consists of everything, whatever we have inside our body, like liver, like digestive system, like circulatory system, like blood vessels, spinal cord, nerves, everything. So internal system consists of all these components. So for the survival of the component of the body system, that is cells, it is depending, so the functioning of the cell depending upon the proper maintenance of the internal environment. Means if there is any infection, see, for proper functioning of the cells, your body should work properly. Means if there is an infection, so because of that infection, you may get some fever will be there, or if there is a imbalance in the salt or imbalance in the ions, so there is maybe some fluctuation in the blood pressure. So for proper functioning of the cell, we have to maintain the internal environment, or we have to maintain, or we can say that the proper maintenance of the internal environment is required. So to maintain the homeostasis, body system also have to take part in homeostatic mechanism. So the body system not only maintaining the internal environment, they allow the body to utilize the external environment for the resources, or for waste removal, like waste removal in terms of waste, different type of, in terms of sweat, in terms of urine, like this. So this is all about the homeostasis. Body system is also a part of this homeostasis maintained. So bodies also have to heal itself. So every organ plays a very important role. Each and every organ present in the body plays a very important role to control the to control the overall health of, or to maintain the overall health of the human body. So this is body system and homeostasis. So target of next is target of homeostasis. So as we know that human body is having different type of system, like digestive system, circulatory system, respiratory system, excretory system, immune system, musculoskeletal system, different type of system. So these all system is actually helping in their own way for maintaining the homeostasis in the internal environment. Means all these systems are responsible for the proper functioning of the human body. Let's suppose if there is a problem in digestive system, it's resulting either in loose motion or in constipation. Or if there is if you have cold and cough, so if that cold and cough is actually results because of that cold and cough, your respiration system will be affected. You will not get the proper respiration. Either sometimes you have to take a deep breath, or sometimes some infection will be there. So because of some infection will be there, the respiratory system will not work properly. So all these digestive systems, or all these systems present in the body, either you will say circulatory system, where the blood is revolving and how blood is flowing in the whole body, respiratory system, how lungs works, the digestive system, where we have a stomach, pancreas, and all these are responsible for maintaining the proper environment. So these are the few points which show the target of the homeostasis. First target is Maintaining adequate concentration of energy sources like fat, glycogen. Glycogen, we have fat present in the system. That is energy, obviously. We have glycogen. When you that I'm going to discuss further also what is the importance of glycogen and all. And the second point is have adequate and continuous supply of oxygen. Certain type of drinks where which are saying that you will which enrich you with some oxidants or antioxidants, or some oxidants should be there. Limiting the concentration of the waste material means it should limit the waste, means whatever we are taking, whatever intake we have taken, it should take maximum minerals, maximum energy, maximum things from that particular intake. Second thing is sustaining the optimal value of pH for proper functioning of the cells, maintaining concentration of the water, salt and electrolytes in the body, regulation of the volume, pressure and temperature of the body within the sustainable range. So now there are certain critical values that should be maintained by by the body itself, like pH. So the critical value for the human pH is should be between 7.35 to 7.45. As you know that water content, our body is having 70% of the water. There should be proper ratio of the water, electrolyte and so on, and the volume. So I will say that in the regulation of the volume, so the critical value of the volume is between 4,500 to 5,700 ml, and the temperature of the body should be maintained around 37 degrees centigrade. And as and the pressure, the body pressure or the blood pressure should be between 120 to 80 degrees centigrade. So these are the some critical values which a body or homeostasis has to maintain for the proper functioning of our cells or for a proper functioning of our body. So these are the few targets that is achieved by the homeostasis. So now, as we can say that homeostasis is actually the main word or is responsible for controlling each and every parameter like volume, pressure, temperature, pH, salt, water level. Everything is achieved or controlled by homeostasis. Now, imbalance. Obviously, there should be some imbalance. Now we are going to discuss what are those imbalance and how this imbalance, what are the effect of this if there is an imbalance in the homeostasis. So how it is going to affect the body? Obviously, our body is one of the most, our body consists of one of the most critical and one of the most efficient adaptive control mechanisms. If you sometimes while cutting while cutting vegetable and all, we see some cuts on our hand, and it heals itself after some time. So that is nothing but a homeostasis. How body will heal itself from different disturbances. If there is any infection, so after some time, body produce antibodies for that, so that that infection will be reduced. We take an example of COVID. If I take an example of COVID, so what will happen? So body produces the antibodies which is used to fight against the virus COVID. So within 14 days or within 28 days, something like that, people are getting cured because they are producing the antibodies, so that they are producing the antibodies against the COVID virus. And also some pharmaceutical industry, you are seeing that now you are reading different type of companies or different labs are trying to produce some vaccine related to COVID. So that is different part. So this is imbalance in the homeostasis. So homeostasis is actually responsible for producing the antibodies which are used to fight against any infection, any virus, any bacteria present in the body. Because these virus and bacteria are responsible for the degradation of the health. So in order to heal itself, the homeostasis process should be fine. So, but still there is some imbalance in the homeostasis. So as we know that all the organs present within the body act together. Sorry. act together through a combination of hormonal and nervous mechanism for maintaining the homeostasis. Let's take the again one example in day-to-day activities that we can say that functions such as regulation of respiratory gases, protection against disease agent, day-to-day activities we are doing food, salt balance maintenance, regulation of energy. These or constant body temperature maintenance. So all these regulations are done by the homeostasis. See, what should be the like we have to measure the body temperature should not be more than around 37 degrees centigrade. Blood pressure should be between 120 to 80 degrees centigrade. So all these things done by the homeostasis. Now, but there are certain but sometimes due to infection or any other reason, there is an imbalance in the homeostasis. So what is that imbalance? Due to the external disturbance, the property of homeostasis gets disturbed and may cause disease in the body. If you catch a cold, let's take an example. Respiratory system is, if you catch a cold, so what will happen? You will have the problem in breathing. There is cold, there is cough every time, so you will sneeze. So there is a problem in the respiratory tract. So due to some external disturbance, like if you eat very cold, ice cream, if you eat ice cream, a lot of ice cream, cold ice cream, then what will happen? It results in it results in an infection. Because of that, you will have cold, and it is directly affect your respiratory cycle. So these are the external disturbances is actually disturbing all the homeostasis, which may cause disease in the body. Due to reduction in the efficiency of the system, as we know that as your age increases, obviously the each and every there is a reduction or there is a aging in organ also, in human organ also. That is also one of the, you can say, the reason for the imbalance in the homeostasis. Second thing is resultant of an unstable environment. So this is very important. This is a very good example. Sometimes you will see if you will go from one environment to another. If you are sitting in a very cold environment in AC, so it is advised before going out, you should switch off the AC, wait for 15 minutes. Why there? If you immediately go into the sun, what will happen? You are actually risking the life. You may catch a cold. Something will be there, some impact because of the imbalance in the temperature. You will get some infection, so which actually increase the risk of illness. So because immediately your body temperature is 37 degrees centigrade, or you are in a very cold environment, immediately go into the very hot environment. So because of the hot and cold, the unstable environment, like this is unstable because you didn't allow your body to stabilize itself. You immediately went out, and you because of that you will get some disease in terms of maybe some people will, if asthmatic patient, they will have the problem of this respiration, or sometimes they have to take respiration because of the lots of humid environment. Also, they can't take too much cold water, too much cold things. So these all things will unstable environment, which is also increasing the risk of which reduces the efficiency of a body, which in turn increase the risk of the illness. Then physical changes, obviously aging. As the age grows, obviously the organ will also ages and the efficiency is also reduced. So this is all about the homeostasis. Now I am going to finally, there is one question that we have to ask from us is, what is how the physiological control system is different from engineering control system. So this is a small table where I have write what are the difference between engine control system and physiological control system. Engine control system are single and predefined tasks can be achieved. Where in physiological control system, it is versatile and capable of achieving different tasks. See how our body is maintained. Our body have to maintain the temperature at 37 degrees. At the same time, pressure should be between 120 to 80. At the same time, pH should be between 7.5 to 7.35 to 7.45. At the same time, it has to maintain the uh, uh, volume around 4,500 ml. So you see our body itself maintaining or achieving different tasks at the same time. But in a gene control system, we can achieve either one or maximum two tasks at a time. Second thing, characteristic of various components of the system are known. We know, see, if I take an example of some process, like chemical process. I know what is going on, what are the factors which is going to affect this particular process. This particular process, what are these parameters? If this parameter changes, so what will be the effect on the output? But in physiological system, in physiological control system, the characteristic of various components of the system are known and difficult to analyze. If I take we can understand like this. Just take the two person. One's age is 20 years old and one person is 50 years. Obviously, if we consider the physiological condition of both the person is different, we can say that the person who is 20-year old is pretty fit in comparison to a person who is 50 years old in a normal condition. Second thing, interaction between the different components is of the system is very less. Means obviously there are all the system interconnection or coupling. The coupling between the system parameters or between the system is between the different component of the whole system is less. But here we see the whole human body is highly coupled system, or we can say there is a high degree of interaction between them. A very good example for that, a person who is suffering from hypertension or high blood pressure. You will see generally if you will take, once a person is suffering from high blood pressure, or once that cycle will come and the person is having this blood pressure ranges above 120. At the same time, what will happen? Heart has to pump blood at a rapid pace. So increasing in the blood pressure will result in increasing in the heart rate. So there is a interaction. Means if a person's blood pressure increases above the some critical value, like 120 is the higher range. Let's suppose if its value is around 150 or 160, so that is high blood pressure. That's why we call that person suffering from hypertension. So that is actually in order to hello. So in order to lower down that the blood pressure, heart have to pump on a more rapid pace. So at that time, if you will check the heart rate, it will be more in comparison to the normal condition. So this we can say that there is a high interaction between the heart as well as in the blood pressure. Second, generally engine control systems are non-adaptive in nature. We have to make them adaptive, either using fuzzy, either using neural, any machine learning algorithm, many artificial intelligence. Generally, if we talk about PID is a non-adaptive or static type of controller. But we can make PID adaptive by incorporating fuzzy, some external incorporation in them. But on the other hand, all these physiological control systems are adaptive in nature. See, if you are sitting in a very cold environment or in an AC at 18 degrees centigrade temperature, after some time, if you switch off the AC, after some time, body will self-regulate itself according to the system, according to the surrounding temperature. So we can say that it is highly adaptive in nature, and engineering control systems are generally non-adaptive. We have to make it adaptive in nature. So now I'm going to take the very good examples from day-to-day life is for physiological control system. One is maintaining the blood sugar level. Maintaining the one second. Yeah, maintaining the blood sugar level. So this is the block diagram This is the block diagram which shows how the blood sugar is maintained in the human body. So as you can see, there are two paths. Path one, there are two paths, path one and path two. So first path is used for if there is an increase in glucose level, and second path is used when there is fall in glucose level. So when you take, let's suppose first we consider path one. Suppose you take one sweet, mithai. In Hindi, if you say mithai, if you take one sweet, what will happen? It actually, that sweet will increase the blood glucose level in the body. Now pancreas works as a sensor. It immediately senses the increase in the glucose level, sorry, the increase or the rising of the glucose level in the bloodstream, and accordingly it will act. How it will act? It produces the insulin. So it releases the insulin and one more thing that is called peptide hormone. Two things pancreas produce. If there is an increase in blood glucose level, pancreas produce, one is peptide hormone and other is insulin. So that insulin interact with the downstream blood cells or the target cells in the body. As you can see, pancreas releases the insulin, and insulin is further interact with the blood cell as well as the liver. So what will happen? So that blood cell is actually in blood cell, it causes the uptake of the glucose from the bloodstream into the cell, and whatever the glucose present in the blood, it actually uptake give it to the blood cells, and whatever the excess glucose remaining, that is stored in the liver in the form of carbohydrate and glycogen. Again, you take the sweet, blood glucose level rises, pancreas senses the there is a increase in the glucose level in the body. It secretes the insulin and peptide hormone. That insulin interact with blood cell and the liver. So whatever the excessive amount of blood, excessive amount of glucose level present in the blood, it give uptake to the blood cells, give it to the blood cells, as well as after that, whatever excess is remaining, excess glucose is remaining in the body, that will stored in the liver in terms of carbohydrate and glycogen. So what will happen? Because of this insulin, it ultimately the blood glucose level decline and our glucose level maintain between 90 to 100, 90 mg per 100 ml. So this is the critical value. Now there is fasting, and that is different. I'm not going to take that, but this is the critical value which of the glucose level should be maintained within the body. So this is the one part. When you take the soup, let's suppose from several hours after eating a meal. So you took a, let's suppose you took a meal at a breakfast at 10 o'clock, something 10 a.m. And after 3 or 4 hours, what will happen? The glucose level will falls. That is sensed by the pancreas. So now what will happen? First, in path 1, the glucose level is above 90 mmHg. So the excessive glucose, whatever is present in the bloodstream, it is either stored in blood cells or in liver. Here, after taking a meal, after a certain time, after 2 or 3 hours, the glucose levels falls down. What will happen? That is also sensed by the sensors, that is nothing but a pancreas in case of human body. It will sense the fall in the glucose level. Then it will direct the liver. As we know that from path 1, as we know that liver, the excessive or the glucose is stored in liver in terms of carbohydrate and or glycogen. So in second part, pancreas direct the liver to break down the glycogen, to break down this glycogen in the glucose monomer. So what will happen? So once that glycogen is break into glucose monomer, so finally the blood glucose level will be rises in the body, and again the glucose is maintained at 90 mg per 100. So this is the best example of the homeostasis, whatever we have discussed, how body. So this is how the body maintains its blood sugar level if there is an excess or if there is a fall in the blood glucose. Now, second example in this scenario is concept of stretch reflex. It is also a very good example. So we can say that it is the most simplest and fundamental example of the physiological control system. So this stretch reflex is actually a routine medical examination in order to assess the status of the nervous system. So what will happen? How it is going to? So this is the block diagram which shows the muscle reflex, and this is the diagram or schematic diagram which shows the how muscle reflexes work. As you can see, when the sharp tap is given to the patellar tendon, this is patellar tendon. When a sharp tap is given to this patellar tendon in the knee, it leads to the abrupt stretching of the thigh muscle. So this is a thigh. When a doctor give a tap on this patella, so what will happen? It reflects or it stretches this the muscle of the thigh, which is called the stretch. Stretch receptor. This is called the stretch receptor. So the muscle spindle or this muscle spindle is also called as stretch receptor. This muscle spindle is also called as receptor, which is activated because of the sharp tap on this patella. So as you can see, once this stretch receptor, which is connected to this patella, get the information that there is a tap in the on this particular patella, it will send the signals through some sensory neuron to the spinal cord. And according to that, in order to balance this upper stretch muscle, again you will see this circular type of signal. So this is the information coming to the spinal cord, and accordingly spinal cord will send the signal back to this spindle or this muscle, that is called afferent muscles, to contract. So these two muscles will contract, and what will happen? The lag will be extended. So the lag will be extended. So this is the closed loop system. This is the how this is the stretch reflex example. Or we can also see this block diagram. So in this block diagram, that this thigh muscle corresponding to the plant or control system, which we are going to control. The thigh muscle is our the system or the plant. The initial stretch produce so this is the external disturbances, or the initial stretch produced in this thigh muscle due to the tapping on the patellar muscle is given by XT. So what will happen? So because of this tapping, there is an increase in the muscle spindle length. So the thigh muscle will increase, and because of that, it will sense there is a feedback sensor. So it will be then So once you get a tap on this patella, thigh muscle will stretch. This information is sensed by the muscle spindle, which is nothing but the spinal cord. So this muscle spindle translate this muscle spindle. This is muscle spindle will connect it to this spinal cord. So this will translate this length in terms of to this spinal cord through afferent nerve. There is one nerve which is used to send this signal, whatever the stretch in this muscles, this afferent nerve is actually sending this signal to the spinal cord, and accordingly, the spinal cord will send the signal to the afferent nerve. This is afferent and this is afferent. Both are different. So this afferent nerve to contract itself so that there is a lift in the or there is a lap extension. So in order so that the muscles so that the thigh muscle will again come to its original position. So this is the one more example of the homeostasis or how physiological control system works. Now we talk about what are the importance of the mathematical model of physiological system. Before going to discuss, there are two statements that I am going to tell you, given by Enrico Fermi and Dennis Noble, related to mathematical model. So, according to Enrico Fermi, there are two possible outcomes. If the result confirms the hypothesis, you have made a measurement. If the result is contrary to the hypothesis, then you have made a discovery. Means if we and second is Dennis Noble, what he said about the mathematical model is we often learn as much as from the failure as from the success of the mathematical model. Means both these statements shows there is the importance of the mathematical model not only in physiological system, in other systems also, simple engineering problem also. So even though you will get success or failure using what are the different terms you can be fail in if you are working on a real-time problem. So with the help of that mathematical model, you can find out the drawbacks of the system. So importance of the first point is it helps us to understand the complex web of interrelation between the various components of the biological system. If I say how blood pressure is related to heartbeat or how heartbeat is dependent on the blood pressure, with the help of the mathematical model, we can easily find that. Second thing, it helps us to gain a better understanding of the system as a whole and in turn predict its behavior in an altered state. Means as you see nowadays you will see a lots of research is going on the development of the mathematical model of the COVID basically SCIR model where people are predicting after 1 month this much will be the persons, after 2 months this will be the persons affected by the coronavirus, how much will be infected, how much will be susceptible, how much will be cured. Like that, they are actually by utilizing this mathematical model analysis, they are predicting the behavior of the coronavirus patients. Like how much will be affected, how much will be cured, and what is the effect of the lockdown. So these all things. Normally, you can't do survey, everything you can't do survey. But if you have the mathematical model, and one more thing, it will give you the insight about the particular problem. It is not mandatory that it will give you the exact solution. But yeah, from the mathematical model, you will have the information, little bit or some information about the system. So it helps to gain the better understanding of the system as a whole, and in turn predicts its behavior in an altered state. Second, the third point is that it enhances our understanding of multiple complex biological processes like enzyme kinetics, metabolic networks, signal transduction pathways, different. And last is the other side of this mathematical model. So mathematical modeling in conjunction with computer simulation had a significant impact on multiple stages in the drug discovery and development process. So that's why these are so the mathematical models related to this method, this drug discovery and development process are called pharmacokinetic model. Pharmacokinetic and pharmacodynamic. So we are going to discuss this on how mathematical models are utilized to find out the different drug discovery, development process and all. So next is mathematical model for drug discovery and development process. On the basis of that, it can be divided into two parts: pharmacokinetic and pharmacodynamics. Now, what is pharmacokinetic and what is pharmacodynamics? So, it is again a prescribed definition, just like we have discussed what is physiology. So, process of an uptake of drugs by the body, the biotransformation they undergo, the distribution of the drug and their metabolites in the tissue, and the elimination of the drug and their metabolites from the body over a period of time. We will discuss what is exactly this means. And second is study of a pharmacological action on living system, including the reaction with the binding to cell constituent and the biochemical and physiological consequences of these actions. So these are the definitions related to pharmacokinetics and pharmacodynamics. To simplify these equations, now what is pharmacokinetics? Pharmacokinetic is these models are called as PK/PD model. PK stands for pharmacokinetics, P K pharmacokinetics, PD pharmacodynamics. So this is the whatever the model is developed using these two terms are called PK/PD model, which is used to develop for the drug discovery and development process. So now what is pharmacodynamics? So pharmacokinetic. So pharmacokinetic is what the body does to the drug. Means, see, as you see, like process of uptake of drug means if you inject a drug into the patient's or into the human body, so how body is going to work how body does to that drug means how much or we can say that like if you inject a person with some dose, some dose, so how body does to that drug, like how the drug is absorbed in the body, distributed in the different part of the body, metabolized. What is metabolism that we will discuss, and how that whatever the excess amount of that grows is excreted from the body. So this is all about the pharmacokinetic. So pharmacokinetic consists of the four important parts. Once you inject a drug into the patient body or a human body, how body what body does to this drug is how drug is absorbed in the body, how it is distributed into the different part of the body, metabolized, how it is metabolized, how it is excreted from the body. Second thing is pharmacodynamics. Pharmacodynamics is what drug goes do to the body. Pharmacokinetic, what body does to the drug, and pharmacodynamics is what drug does to the body. Means after injecting that dose or drug to the patient body, after some time, 1 or 2 or 3 times, what are its side effects? What are its side effects? That can be easily given by the pharmacodynamics. Very recent example, as you are seeing or you are going through news channel, almost 20 or 30 labs or 140 or 160 labs, different labs all over the world are developing vaccine for the COVID. So what they are doing, all these doses or these drug or these vaccines are going in a trial period. So what they are doing, they are developing a drug, injecting into the healthy person, give them a time, gave them a time, so that after some time they are checking what are the side effects of that particular drug, what are the reason or what parameter of the body is actually more affected by that drug, so that this particular side effect will occur. Another example, you know that cancer, all the cancer drugs are cytotoxic or highly toxic in nature. What are the side effects? That hair falling. Sometimes hair falling is there, fever is there. So these are the small symptoms or side effects of the cytokine. Or toxicity level will increase. Patient will not feel hungry. So these type of things, too much dehydration. So these are the side effects of that cytotoxic drug. So if I wanted to model that particular thing, a particular vaccine drug into mathematical form. So that is called as PKPD model of pharmacokinetics and pharmacodynamic model. So pharmacokinetic is what body does to the drug, and pharmacodynamic is what are the effect of the drug after the absorption and everything on the body. So this is all about the PK and PD. Now, effect of drug. So once the drug is injected into the body of the patient, so a drug effect is often related to its concentration at the site of action. So it would be useful to monitor the concentration. So very important point here. The problem initially is that how much amount of drug dose should be given to the person, or we can say that what should be the concentration should be maintained around that site of uh, action, and also we have to monitor the concentration also. So let's suppose concentration should be maintained around 12 mg per ml. That we will discuss in two examples. And the second is receptor site of the drug are generally inaccessible to our observation and are widely distributed. Means the effect of the drugs is once the drug is injected into the patient body or in patient body or human body, so it is circulating in, it is distributed all over the body. So in order to find out in order to observe the effect of that particular drug on the human body, we have to go through different stages, different systems. So what is the effect of the drug on the digestive system? What is the effect of drug on that circulatory system, on the respiratory system? So this is the very almost inaccessible type. Therefore, direct measurement of the drug concentration at the site is not practical. So it is not practical. So we can't find out if we can't directly measure the drug concentration around that particular site of action. So we can measure the drug concentration of blood or plasma, urine, saliva, and other easily sampled fluid because of kinetic homogeneity principle. So now, in order to check that we can see, if you inject a drug, it is not possible to simultaneously take care of each and effect of that drug in each and every system. Like what is the effect on circulatory system, what is the effect on this. So these are some tests. Like so that's why doctor prescribe you to go for blood or plasma test or urine test or saliva test or other easily sampled fluid. Next is relationship between plasma to tissue drug concentration. So there is a linear relationship between whatever the drug concentration you will inject, same plasma drug concentration will be. So there is a linear relationship between the plasma and tissue drug concentration. This is also one good example. This is how prescribed doses will be given. Then it is undergo pharmacokinetic stage. Means absorption, elimination, drug interaction, drug metabolism will be there in this stage, and then it reaches to drug at the site of action. After that, the stage after that we will see the after effect of that particular drug that comes in the pharmacodynamic stage, where drug receptor status, genetic factor, tolerance, everything. So this is also a block diagram representation of a relationship between PK/PD. This we have already discussed. So this is the schematic depiction of pharmacokinetic. Both are same. So this is the one circulatory type of representation of the particular how pharmacokinetic works, and this is the schematic diagram, block diagram. So as you can say, there are four important stage of pharmacokinetic: absorption, drug distribution, metabolism, and drug elimination. So metabolism, what is metabolism? Metabolism is nothing but which describes all chemical reaction involved in maintaining the living state of the cell. Whatever the chemical reaction, whatever the chemical imbalances reaction going inside the human body, it is actually it describes that thing in order to maintain, which are responsible for maintaining the living state of the cell and organism. It is of two types: catabolism and anabolism. That is what we are going to discuss. So this is how absorption. Once drug in the dose form is given to the patient, or drug in the form drug in the form of dose, oral or in terms of injection, it will reach to into the bloodstream where it acts as a free drug. So it will interact, it will be distributed into the different part of the body. Like it will be the drug is connected, is goes into the site of action, or maybe some storage tissue it will go, it will interact with protein binding, or some biotransformation is there. Biotransformation is nothing but a chemical alteration. So free drug will result in chemical alteration in the body, which produce the metabolites, or finally we have this excretion of that particular free drug in terms of urine, bile, sweat and saliva. So this is the depiction of that whole pharmacokinetic process. Now, pharmacodynamics we have already discussed, like relation between drug concentration and site of action. What are the after effect of that particular drug? So next is compartmental model. Now we are going to discuss what are the compartmental model for what are the compartmental model for pharmacokinetics. So generally it is also called as empirical model. It is a traditional and most commonly used approach to pharmacokinetic characterization of the drug. So these models actually simply interpolate the experimental data and allow an empirical formula to estimate the drug concentration with time. So these compartment models are of two types, are divided into two categories. One is mammillary model and catenary model. So we are going to discuss the mammillary model. So mammillary model, and these are the few assumptions that we have to take before developing any compartment model. First is that the body is represented as a series of compartments. Second, each compartment is not real; it is hypothetical in nature. Hypothetical in nature. Second thing, the third is within each component, the drug is considered to be rapidly and uniformly distributed. Means first here you inject the drug and it is uniformly and rapidly distributed and all over the compartment. Third, the rate of the drug movement between the compartments is described by the first order guide. Means if the drug is moved from one compartment to another compartment, so that the movement is actually denoted or described by the first order kinetics. Finally, the rate constant are used to represent the rates are used to represent the rate of entry or exit from the compartment. So this is the mammillary compartment. We just come to this block diagram of this mammillary compartment. So these are the 5 models, and according to that, according to compartment, we have this model 1, which is also called as one compartment or open model. Intravenous administration. If drug is directly injected into the bloodstream, so that is called intravenous administration of the drug. So it is having only the whole body is considered as a one compartment. So that's why it is called one compartment open model. So this is intravenous, and this is a one compartment open model. Extravascular means if a dose is given through some oral dose, like tablet, in terms of tablet and dose is given, so that comes into extravascular administration. This is again the body is considered as a one compartment, not two compartment open model. As you can see, there is one and two compartments. So this compartment is called as central compartment, and this is called as peripheral compartment. And there is a rate, as we see here. We have discussed like rate constant. What rate? At what rate? See, at what rate this particular drug dose is moved between the one compartment to the another compartment. Simply with this is for intravenous, this is extravascular. So this is for two compartment. I mean 3 compartment. We have one body is divided into 3 different compartments. And like last is 3 compartment extravascular. So we are going to discuss how 2 compartment model is going to. So this is the block diagram for the 2 compartment extravascular administration. So, as we see the pharmacokinetic two compartment model in this particular picture, as we see that two compartment model is divided a body into two parts, central and peripheral part, central compartment and peripheral compartment. Whereas central compartment consists of the plasma and tissue. Uh, Plasma and tissue which is further distributed, distribution the drug into the other sorry yeah so central compartment consists of the plasma tissues where the distribution of the drug is going on, or we can say is practically instantaneous. Immediately there is a distribution or in this compartment. And the peripheral compartment which consists of the tissues where the distribution of the drug is slower. So in central compartment the distribution of the drug is quicker, whereas in peripheral the distribution of in peripheral compartment the distribution of the drug is slower. This is GI tract, that is called gastrointestinal system. It is composed of digestive tract, accessories organs, salivary gland, pancreas, gallbladder, and liver. So this is a two compartment model where AG1, A1, A2 are the amount of drug in gastrointestinal tract, central compartment, and the peripheral compartment respectively, where K, K12, K21 and all these are the first order fractional rate constant for absorption, distribution, redistribution, and elimination. So let's take an example, how we administrated the drug into the patient body. So in case of oral administration at time is equal to 0. So the amount of drug in the central compartment, and peripheral compartment is 0. Initially we assume that there is no drug concentration or drug present in the central and peripheral compartment. Whatever the drug or effective dose, whatever the drug dose is, it is present in the gastrointestinal tract. So AGI, that is represented by AGI0, which is nothing but the multiplication of D into S into F, where D is the administrative dose of the drug, S is the salt factor, and F is the bioavailability factor. Now D drug, we know that dose, D is the dose drug of dose. Dose of the drug. So salt factor is nothing but a fraction of the administrative dose that is made up of pure drug, and F is the bioavailability factor, which is nothing but a fraction of dose that reaches the systemic circulation. Means salt factor is it is depending upon the drug dose, the pure drug dose, and F is bioavailability factor, which is actually which shows the how drug doses reaches to the other part of the body. So the amount of drug in GI. So as we see from this particular graph, there is a transmission or there is a movement of a drug from GI tract to the central compartment. So obviously, if there is a movement of the drug dose, so the rate of change of the drug dose, the concentration of the drug dose in this GI tract is decreases. So this is how, what will be the now if I wanted to find out the rate of change of this AGI in this GI tract will be given by this. So as you can see, you all know that free body diagram. Simply, if I wanted to write the rate of change of this GI tract is depending upon the rate at which it is going out from this tract multiplied by its concentration. So rate of change of AGI is equal to minus K, sorry, this is K, KA AGI into T. That means this rate constant multiplied by the concentration present in the or drug dose present in the GI tract. Now if I wanted to find out the concentration, this is all about the drug dose present. So concentration present inside the central, I wanted to find out the rate of change of drug concentration in the central system. So that A1 is, what is A1? A1 is the drug dose. A1 are the amount of drug present in the GI tract, central and peripheral compartment. So now the drug concentration. If I wanted to find out the concentration of the central compartment, that is nothing but the volume is V1 of this whole. Let us consider the volume of this whole compartment is V1. So A1 divided by V1 will give me the concentration C1. So the rate of change of C1, which is nothing but C1 is nothing but A1 divided by the volume V1, is equal to this K0, this rate constant, this the concentration is given again the free body diagram. All these two are incoming, these two are outgoing. Accordingly, we have the signal. Whatever the incoming is represented by the plus sign multiplied by their concentration, and whatever is outgoing is shows the negative point multiplied by their concentration. So concentration in the central compartment is C1. So I can say that the rate of change of concentration in the central compartment is equal to minus K10 into C1 minus K12 into C1 plus K21 into C2. This is nothing. This is coming from the peripheral compartment. So this is the K21 into C2, and finally K1 into Ka. Ag1 upon V1 is you can say CL also. CGI, the concentration of the drug present in the gastrointestinal. The same we can for peripheral. So this is how we can develop a two-compartment model, pharmacokinetic two-compartment model. If we integrate, so this will be integrate both these two equations, we get a final concentration. So now the last two is chemotherapy model and how to do meta simulation. So this so this is a two-compartment model. Two-compartment chemotherapy model, which consists of a proliferating cells, also called as cancer cells, and quiescent cell. Quiescent cells are the equilibrium cells, which are on the verge of converting into proliferating of cancer cell. So A is nothing but the rate of change of the cell into proliferating. M is rate of change of this proliferating into quiescent, and D is the rate at which quiescent cell changes into proliferating cell. So these are the equation. We show the PKPD model. So this compartment show the effect what this compartment does onto the body, and this show the PD model like pharmacokinetic. The after effect of this drug, if you inject the drug, it will affect the cancer cell as well as the quiescent cell. And what are its after effect? After effect is decreasing, it will affect the normal cell concentration of the normal cell as well as increasing the toxicity. So this shows the PD model and this shows the PK model. And the relationship between the rate of cell killing and drug concentration given by this equation. Finally, now steps of simulating simulate this on MATLAB's simulation environment. So first is open the MATLAB and create a new simulink model. This is how you will get the simulink model. Second step, open the simulink library and select the atomic subsystem block model and add it to the model itself. So this is how we have this is simulink library. We have to open we have to click on this simulink library, which results which open like this, and we type in such atomic subsystem and add this atomic subsystem into the untitled model. So open the subsystem and you will see 2 points for input and output. Create extra point for the output because we have the 5 output parameter in the model. So this is how this subsystem looks like. This is output and this is input and this is output. Copy paste. This is 5 because we have 5 outputs, so we copy paste as 5 output. Now again click on the simulink library and select the block as per the equation and arrange. So these are 2 equations for P and for Q. Now how to simulate this P and Q? So this is the atomic subsystem for which corresponds to dP by dt, and this is the atomic subsystem is corresponding to dQ by dt. Now inside how we can arrange. So this is the simulink. We see A, M, and as we can see the equation, we have constant value A, M, and B, and GT is depending upon the drug concentration as well as GT is nothing but S1 into dt. So we simply take the constant value according to their sign, sum it, subtract it, multiply it by PT, Similarly, QT is quiescent cell, the input from the Q compartment multiplied by B sum. Similarly, GT will be multiplied by PT, and finally we have this. We have to integrate this so that we can get the PT. So finally, we have our proliferating cell. So whatever the effect of the drug concentration, this is how you can simulate a linear differential equation in a MATLAB. Similarly, we can simulate for QT. Now, repeat this process till all the equation will be designed, and the final simulating model will be like this. So this is the open loop response. This is how the drug concentration changes the toxicity. If concentration increases, obviously the proliferating cell or the cancer cell and the quiescent cell will decreases, and the after effect is decrease in the normal cell. Now PID control scheme. This I'm not going to. This is only I'm going to discuss. This is the last I'm going to discuss. So this is how if I wanted to similarly search the PID block from the simulation library and add it to the model. We have the model, and from closed loop control structure, we have this closed loop control structure in order to control the drug concentration. So again, click on this PID controller, PID parameter. You will have this block. Now, in order to tune, click on this auto tune button, and it will gives you the best optimized parameter for this particular problem. So finally, we have our controlled output as follows. So this is how we have this controlled drug concentration. The reference input is 12.0. Similarly, this blood pressure control. If, Dr. Verma, hello. Hello. Yeah, Dr. Jain. Hello.
Hello, Dr. Nikhil, I can hear you.
Sir, time is, I will just 5 minutes I will take, and then I will complete. I think it is almost a time.
Yes, sure. We can take a 5 minutes break.
Okay, 5. No. Just I'm going to complete it. It almost 9.
You are going to complete in 5 minutes.
5 to 15 minutes, because the time is.
It is okay, sir.
2 to 3:30 was the time. So accordingly, I prepare my PPT.
Yes, it is
so
fine,
now,
sir.
So blood pressure control. So another example is blood pressure control. So this is MAPP model. Similarly, we will have this MAPP model, one drug infusion pump and a control. Same closed loop. This shows this closed loop diagram for MAPP model. Output is being sensed by the sensors, which is again compared with the reference blood pressure. It produces an error, and according to that error, controller will take the action. How much amount of the drug is to be injected into the patient in order to normalize the blood pressure. So the modeling of the response of a patient to the effect. This is SNP. SNP is nothing but a name of the drug which is used to lower down the blood pressure, the value of to control the blood pressure during surgery and all. This is called sodium nitrocrucide. This SNP is nothing but sodium nitrocrucide, which is injected into the patient during any surgery. There is a fluctuation in the blood pressure. So this is the transfer function model. In previous example, we took differential in terms of differential. Now here we consider a transfer function model. So now the patient model dynamics has been taken like this. PI is drug sensitivity, PI is the initial transport delay, TC is nothing but a recirculation time delay, recirculation constant, lag time, and all. These are the some parameters. So we have three according to the patient survival, or according to the patient efficiency or patient physiological condition. We divide this particular model into three terms: nominal, patient model, sensitive, and insensitive. And accordingly, all the parameters will be varied. So MATLAB simulating. How the MATLAB simulating diagram? So this particular GS, this model can be again we can constitute or we can develop in MATLAB simulation environment, and it will be like this. These are nothing but a transport delay. Everything you just click on this Simulink library, whatever you wanted to find, like transport delay, you just write into the search doc, go to the search doc, write transport delay, and it will give you the transport delay. And click on that, right click on that particular doc, and add to the model. Now there is one more disturbance model that we have considered for this particular. This is the patient model. Sorry, this model is the patient model, which is shown like this, and we add disturbance in this model. So this is the disturbance model that has to be considered, that was considered. Now how PID flow? Similarly, so we have this patient model that I already showed you. Then disturbance model, we have that the reference BP blood pressure value. So this patient model can be changed for normal, sensitive and insensitive. This can be changed according to that particular model parameter values for normal person, for sensitive patient model, for insensitive patient model. And one disturbance model is also included. So this is how, again, the same we designed the PID for chemotherapy model. So this is how we can design the PID for blood pressure. Again, click on this PID block, you will get this block. Tune, click on the tune, you will get this parameter. Now you are seeing the negative value of KP and PI. It is physical, because if your system is negative response system, then the value of the KP and PI should be, you will get the negative. We will see the reference. Let's suppose for this particular problem, I wanted to maintain the reference at minus 30 mm, roughly minus 30 mm Hg. So this is how the blood pressure will work for minus 30 for normal patient, PID parameter for normal patient. This is for sensitive patient model. So every time you will see the PID controller is able to maintain the blood pressure at its reference point. This is how we can design our processes. So these are the differences. Thank you. Any queries?
Yes, audience, if you have any queries, you can please ask Dr. Nikhil.
Please, one by one, you can on your voice and you can ask. So I got
Someone has
this Rocha Pramanik. Can the MATLAB model be used to detect the fluctuation in blood pressure? See, we can do lots of things using modeling or MATLAB model. Like yes, it can be blood pressure fluctuation. Accordingly, you have to fluctuation. How you can see? I will tell you how you can check the MATLAB fluctuation. This is the parameter. We have this ID. This is the open loop system. If you change this ID, accordingly you will have change in the blood pressure. So this is output upon input, which is given. This is output upon CS upon RS, output upon input given by this GS. So whatever change in the input, it will be multiplied by this GS, and the effect of that change in the input you will get in the blood pressure. So yes, you can check the fluctuation, what are the maximum minimum range of the input, so that it will be within the range like 120 to 30 degrees. So what are the value of the input that blood pressure is maintained at 120 degrees, 120 mm and for what value of the input the value or the blood pressure is at 80 degrees. So you can check the fluctuation of blood pressure by changing the input using the MATLAB model. Any other?
It seems there are no more questions. Dr. Nikhil, we can close the session, I think.
Okay, okay, okay.
So thanks a lot for your wonderful presentation.
Thank you, sir.
If in audience, if anyone has any further queries, you can get back to Dr. Nikhil at his email ID.
Sir, Karan sir will share my email ID, and if you have any queries, you can mail me. Okay, sir.
Yes, sir.
Okay, sir. Thank you. Thank you very much.
Have a nice one. Okay. Thank you, audience. So see you tomorrow at 10:50 a.m. So tomorrow the first talk would be by Dr. Milli Pant from IIT Roorkee. Her talk would be centered around optimization tools. Okay. Have a nice time.